In looking at cell division, there's two main types that we're going to care about. The first one is mitosis. It's a type of cell division that enables cells to replicate exact copies of themselves so that as you grow and get larger, you need to produce more cells. When you have damage to any tissue systems, like you skin your knee or something, you've got to replace those cells. And also some organisms use it for reproduction. Meiosis is the second part of cell division, and in meiosis you're forming sex cells, so you're making sperm cells and egg cells. Those are called gametes, and they're used for sexual reproduction, and they have half the number of chromosomes of the original cell, so that when they combine to form a zygote, that zygote is going to have a full set of chromosomes. Looking at the cell cycle, there's a series of events that are initiated. Most cells are actually just in a period called the G0 phase. In the G0 phase, the cell's just doing its normal job. So if it's a muscle cell, it's contracting and relaxing. If it's a nerve cell, it's sending sensations and so forth. But they can be triggered out of the G0 phase periodically into this version of the cell cycle, which consists of three main phases. You have the G1 phase, in which the cell grows and enlarges to prepare for a duplicate set of DNA. And there's a checkpoint here that tells cells when it's time to move beyond that point. And if these checkpoints throughout this diagram get messed up, then you can actually have cells that are growing uncontrollably, and that's what causes cancer. So the next phase of the cell cycle in a normal healthy cell is called the S phase, which is called known as DNA synthesis. So in this stage, you're actually replicating your DNA, and there's more on that in another unit, how DNA is actually replicated. Then we go into what's called the G2 phase. In the G2 phase, the cell is getting larger and preparing so that when it splits in half, it'll be the same size as the original cell. And there is a checkpoint at the end of the G2 phase that says, okay, everything's preparing for the mitotic phase, which is a series of interactions in the cell that will actually result in the overall division. And that's where we're headed in the next few slides. So this is an overview of mitosis. In mitosis, there's four main stages. So we come out of G2, this precedes mitosis. And the cell has already replicated its DNA at this point, so it has two sets of DNA. The first part of mitosis is called prophase. And prophase, you've got a number of things occurring. First, you have what's called the spindle apparatus forming, and that's going to attach to the DNA and move it around the cell. You have chromosomes condensing to form sister chromatids. So that's, again, a process that we'll talk about in another unit. How that actually works. But for today, you just need to know that they actually supercoil and go from this long, stringy looking stuff down into a tight chromosome, which looks more like a line. And then we have the nuclear envelope, which protects the DNA, breaking apart so that you can move the DNA around the cell. The second stage of mitosis is called metaphase. And in metaphase, the spindle apparatus has microtubules that connect to the chromosomes. They connect to what's called the centromere of the chromosomes. The ones that connect are called kinetochore microtubules, and they connect to the centromere of the chromosomes. And then there are non-kinetochore microtubules that go between or around those chromosomes. They don't actually touch the chromosomes. They simply connect to the other part of the spindle apparatus to enlarge the cell and push it apart. But the main thing is that the chromosomes line up on what's called the equatorial plate, like the equator of a globe. It's also known as the metaphase plate. And they line up in such a manner that when they go into the next stage, called anaphase, they get pulled apart, and you end up with one set of chromosomes, one set of DNA, heading towards the north pole of the cell, and a second set of DNA or chromosomes being pulled towards the south pole of the cell. This is separating the DNA so that the two copies will be pulled to opposite ends of the cell so that you can get to the last stage called telophase and cytokinesis, where you get a new nucleus forming around the two different sections of DNA, and you get the cell dividing in the middle to form two new cells, one that is an exact replica of the original cell. Looking forward, we have slides that go into more detail, and so if you come into these slides and you have questions about what anything means in here, please send me an email so we can talk about it. Here's an example of a tumor that's formed in lung tissue, so this would be lung cancer, and you can see the mass here on the right lung in this abnormal chest x-ray. And over here in a normal chest x-ray, that mass is not there. So this individual here has a tumor, which results from uncontrolled mitosis, a cell dividing and dividing and dividing, forming more and more cells. And they're not going through the appropriate checkpoints, and they're not stopping and checking their DNA. So they're really not functional cells, and it creates problems for the body. Meiosis is a bit more complicated. It's actually not too bad. It just goes through two separate divisions. One is called meiosis 1, and the second one is called meiosis 2. There is only one duplicate set of DNA. It doesn't go through another replication. So what you do is you take a cell that had the original amount of DNA, and prior to meiosis you double that, so it now has twice as much DNA as a normal cell. You then split it in half, giving two cells, and that's what you see over here, that have the same amount of DNA as the original cell, and then you do a second round of division without replicating your DNA, so that you split each of those two cells into two separate cells, ending up with 4 cells that had half the amount of the original DNA. So this is what's used to make sperm cells and egg cells, because as you know, after sexual intercourse, the sperm and egg cell fuse together so that those half sets form a full set of chromosomes. Those cells are called haploid cells because they have half the number of chromosomes, or half the amount of DNA of the original cell. Looking at meiosis, I want to go back to prophase 1. It's the key phase. It's the one that has the biggest difference between mitosis and meiosis. And there are 4 things that happen in prophase 1 which are important to remember. The first is called synapsis, and you can Google the term, but it's a term that refers to the alignment of homologous chromosomes in an order that their traits will be able to be aligned. So that if you've got a chromosome that has the eye color characteristic, you want to make sure it lines up appropriately with the second chromosome that has the eye color characteristic. So that lining up is called synapsis. Then the chromosomes are actually linked together in what's called tetrads. The prefix tetra means 4, and a tetrad actually has 4 strands of DNA. The original chromosomes, there's 2 of each type of chromosome. You have one from your mom and one from your dad for every characteristic. So you got an eye color gene from your mom and an eye color gene from your dad. And actually, eye color is polygenetic, meaning there's lots of different genes, but just to simplify for this example, you have one from your mom and one from your dad. When you get replication of that DNA, you then end up with 2 from your mom and 2 from your dad. When those join, 2 and 2 gives you 4, hence the word tetrad formation. So we have synapsis, which is the lining up of chromosomes. Then we have tetrad formation, where 4 strands of DNA actually get linked together by what's called a centromere. You then have chiasmata formation, and that sounds like it starts with a K, but it's actually C-H-I, chiasmata, and that means that the DNA will actually wrap around other strands of DNA within the same tetrad so that you can exchange the alleles on the different chromosomes. And that leads to the last part, which is called crossing over. And crossing over is the physical exchange of DNA from one strand to another. So again, in prophase 1, it's the phase to focus on in meiosis. 4 things happen: synapsis, tetrad formation, chiasmata formation, and crossing over. That does not occur in mitosis. So here's a slide that compares mitosis and meiosis. The top is mitosis, a skin cell. So whenever you scratch a part of your body, you're actually pulling off skin cells. And so you'll make sure you replace those skin cells by the process of mitosis. And then the bottom is meiosis, and so we end up with, this is showing the formation of gametes, but it really could only be sperm cells, because we'll learn about how females produce only, they only produce one egg cell per month in a monthly cycle called the menstrual cycle. And so their formation of those eggs and release of those egg cells is a little bit unique. But here we've got four gametes, which means it's a sperm, the formation of sperm cells. More on that in the reproduction unit. Here's a diagram showing crossing over, because it is a bit confusing. We've got the green chromosome representing the one from your father, and the yellow representing the one from your mother. You'll notice that they look like X's, and that's because if you trace one of them, it has already been duplicated. So we've got a second one from your father that you duplicated in your cells, and you've got your original one from your mother, and the second one that's been duplicated by your cells. So the green and the yellow are considered homologous chromosomes, and because they have identical strands of DNA, the green is identical to the other green because they are replicated. Green and green are considered sister chromatids. Yellow and yellow are sister chromatids. But green and yellow are not sister chromatids. They are homologous chromosomes. I know that's a lot of vocabulary, but I just wanted to try and give you a brief introduction to this. In looking at crossing over, the first thing we have is what's called chiasmata formation. So this location where they're wrapping around each other, that's called a chiasma. And when you have more than one, here this one has two, where they're actually overlapping, that's called chiasmata, so it's the plural form. And when they actually exchange their DNA, they end up with one strand that has a green arm and a green and yellow arm. The other one, the short end, has green and yellow, and the long end has all green. And you'll notice, the yellow ones as well, that the strands of DNA are now different. So what began as identical strands ends up as very unique strands of DNA. And that's the process of crossing over, the physical exchange of sections of DNA. This is a chart showing you the differences between mitosis and meiosis. And you can review this and ask me questions. I want to talk briefly about a karyotype. So you can get genetic testing on babies or on fetuses before they're born, and you can get genetic testing of your own cells if you gather cells from your cheek, if you swab it or so forth. And your DNA looks like this in these chromosomes, and you get what's called a karyotype, which is an image matching the chromosomes that are the same chromosomes, homologous chromosomes. You'll notice that that this karyotype is actually not of a human. This is of a different animal. And I know that because humans have 23 pairs of chromosomes, and this one actually has 27. There's 26 numbered pairs, and then there's the gender chromosomes, giving you a total of 27 pairs. So this is an animal other than a human. Humans have 23 pairs of chromosomes. This one is a karyotype of a human, and we've got 22 pairs plus the gender chromosomes, which gives you 23 pairs of chromosomes. The gender of this individual, they have these large chromosomes here. Those are X chromosomes, and there's the absence of Y chromosomes. So this person is XX, making them female. Additionally, they have 3 chromosomes at the 21st position, which is going to lead to a condition called trisomy 21, more commonly known as Down syndrome. So karyotypes are pictures that can show genetic abnormalities. Here we've got another image of a human. I know that because there's 22 pairs of chromosomes and then gender chromosomes here. This individual has one X chromosome, but no Y chromosome, and not the additional X, not a pair of X's. So this is called a monosomy. This previous slide is a trisomy. This slide is a monosomy.